Hi, I'm Hannah. I'm a senior and I'm majoring in biology and zoology. This is my first year in the Wyoming Research Scholars Program, and my mentor is Dr. Amy Christ. I'm working on a research project with graduate student Elle Wimmer, where we will analyze the effects of a novel crayfish predator cue on phenotypic plasticity in physid snails. So here's a bit of background information regarding the project. Other studies have shown that snails will exhibit phenotypic plasticity in response to the presence of crayfish predators. Crayfish prey on snails in two different ways. They can either reach through the opening of the snail's shell and pull out the soft tissue, or they can crush the shell and get to the tissue in this way. When snails receive chemical cues that crayfish are preying on other snails nearby, it may result in phenotypic plasticity. The snail could develop a smaller shell opening or a thicker shell to make it more difficult for crayfish to prey on them. So for this project, we will compare the differences and variation of traits between snails from natal environments and novel environments. In this case, the novel environment will be the habitats that contain crayfish. So we'll have 3 populations from locations that have crayfish and 3 populations from locations that don't have crayfish. And then all 6 populations will be exposed to both the novel environment and the natal environment. The snails from habitats lacking crayfish that are exposed to the novel chemical cue, which in this case is the cue from the crayfish, should show higher variation in trait values. This is because these snails grew up without the selection pressure from the crayfish. It was missing in their home environment, so the traits associated with the selection pressure, like shell morphology, were not expressed in their home habitat. In contrast, the snails who originally grew up with crayfish around should show less variation in the related traits because selection acted on these snail populations in their home habitat. So right now, we are currently rearing the snails in the lab so that later we can study the effects that the chemical cues have on the development of offspring from the different populations around Laramie. And then later we will conduct reciprocal transplants and we'll be measuring growth rates and shell morphology every couple of weeks. This will tell us if the novel environment, which is the one with the crayfish, induces increased variation in phenotypic plasticity, and if phenotypic plasticity is different in populations from the areas with no crayfish versus those with crayfish. Thanks for watching.